Okay, welcome to today's session. We'll be doing Re-NEET Biology session where we'll be revising each and every chapter with MCQs. So we'll start with Human Physiology with 60 MCQs each in each chapter, so human respiratory and cardiology. We'll be revising the important diagrams and points through these MCQs and we'll cover the entire chapter through it. Okay, so we'll be starting with the first module of the respiratory system, and the section first will be starting with the respiratory organs and the anatomy across the species. So with that, we start with the first question. Match the organisms. Match the organism with its correct respiratory structure. Okay. Earthworm, they have given gills. Okay. Let's write it down here. Earthworm, A. Insects, B. Fishes, C. D. Mammals or birds, they're given tracheal tubes. Okay. First thing we know, earthworms, they breathe, their respiration is not through gills. So we know the first option. So wherever A with our option 1, cancel it off. Okay. There is A with option 2, moist cuticle. Okay, that is correct. A cannot be option 4, so let's cancel option 4 out. We have two options with A and C. Okay. Now let's look at next option. Insects. Okay. They have given insects with moist cuticle. We know it is wrong. So is there anyone with B too? No. Okay. So insects, they breathe through tracheal tubes, correct? So option B with 4, yes. So we can say that option A is correct. With this we can do fishes. We know it is gills, correct? It is gills. And the third one, the obviously birds and mammals are through lungs. Okay, with this we cover the entire the species across the species. We just learned the basics. So earthworms, they exchange the gases directly across the vascularized moist cuticle, and insects have a utilized network of tracheal tubes that deliver the air directly to the cells. Fishes, obviously, they have the specialized vascular structures called gills. That is called as brachial respiration, and birds and mammals have this highly vascularized bags called lungs. This is known as pulmonary respiration. This is pulmonary respiration. Okay. Now, the next question. Which of the following groups of animals respire exclusively through bronchial respiration? So, what is bronchial respiration? That is through gills. Okay. So, we have to first, the main thing about doing MCQs is cancelling out options. We need to know that which does not have, which has. Okay. So, we have to first cancel out options. In this, which is the most strange one in the options? Obviously, it's non-cilintrates. We have non, we have cilintrate sponges, flatworms. They're all they are all not mammals. They are not uh, part of the. They are non-chordators, correct? They are non-chordators. How can non-chordators have bronchial respiration? Correct. So directly we can cancel out option D. Centipede, I mean insects, centipedes, millipedes. We just learnt about it now. Tracheal tubes, correct? So that is also cancelled. Now we have fishes and amphibians. Obviously, fishes have gills, correct? Mammals does not have gills, so directly cancel off the option. So option A, fishes, aquatic arthropods, and aquatic mollusks. No, bronchial is done by aquatic organisms. Okay. Now, the cartilaginous flap called epiglottis and prevents the entry of food into glottis, gullet, pharynx, esophagus. Now, the main thing you need to know is larynx houses the glottis. So, what is glottis? It is the opening of the windpipe. Okay. So, basically, what happens is during swallowing, this thin elastic cartilaginous flap, which is epiglottis, it covers the glottis to prevent the food particles to enter into the respiratory tract. Okay. So, what is Pharynx, it can't be. Esophagus is food pipe. Okay. What is the difference between glottis and gullet? Gullet is actually the opening of food pipe. Okay. It is the opening of esophagus. So it can't be glottis. Glottis is the opening of the windpipe. Okay. Next question. Up to which structural level are the conducting passages of the human respiratory tract supported by incomplete cartilaginous rings? Okay. Primary bronchi only, secondary and tertiary bronchi only, terminal bronchi, respiratory bronchi, and alveolar ducts. So, the thing you need to know is there is incomplete hyaline cartilaginous rings. Okay. So, what are these incomplete cartilaginous rings do? They support the trachea. Okay. They support the secondary trachea, the primary, sorry, not the trachea, the primary bronchi, the secondary bronchi, the tertiary bronchi, the initial bronchioles up to the terminal bronchioles. We need to remember that. Why is it supporting till the terminal bronchioles? Okay, this is because once the air, this is to prevent the airway from collapsing. Okay, basically due to the negative pressure of inspiration. Okay, so these structural rings, they are not there in respiratory bronchioles and alveolar ducts. Where is it not there? In respiratory tracts and bronchioles, alveolar ducts and respiratory bronchioles, it is not present. Okay, whereas it is present in trachea, primary bronchi, secondary bronchi, tertiary bronchi, and initial bronchioles up to the terminal bronchioles, but it is not there in respiratory bronchioles and alveolar duct. So, what was the question? Up to which structural level are the conducting passages of the human respiratory tract supported by incomplete cartilaginous rings? So, what is the answer? Terminal bronchioles. Okay, this obviously it is not there, so we can cancel it out. We'll get confused and sometimes we mark the second option. No, it is still terminal bronchioles. Okay, the next question. The structural site where actual diffusion of O2 and CO2 takes place between blood and atmospheric air is option A, trachea and primary bronchi. Option B, terminal bronchioles. Option C, alveoli and their thin ducts. Option D, nasopharynx and larynx. Okay, so you need to know respiratory tract is divided into two parts. What are the two parts? One is the conducting part. Where is the conducting part? Conducting part is from the external nostrils till the terminal bronchioles. Okay, but where does the respiratory or the exchange part happens? It is basically happening in the alveoli. Okay, it basically happens in the alveoli. Okay, and alveoli is the actual site for passive diffusion of gases down their concentration gradients. Okay, so what is the answer? Alveoli and their thin ducts. Okay, now next question. The human lungs are situated anatomically in an airtight chamber known as. This is very easy, but suddenly when we see this, we get confused between pericardial and thoracic. How is that possible? Directly we can cancel out abdominal and pelvic. But in the tension, we think, is it located in the pericardial or thoracic? It is located anatomically in the thoracic cavity. Okay, because the arrangement is critical because any change in the volume of the thoracic cavity is directly mirrored in the change in the pulmonary volume. Okay, which is the foundational mechanic required for breathing. Okay, so obviously, where is the thoracic cavity? Very easy question, direct question, but pericardial, I don't know. Sometimes when we think everything suddenly with all the questions in our head, we tend to get confused. Okay, no problem with that. Now, the thoracic chamber is formed dorsally and ventrally by what? First thing you need to know is what is dorsal and what is ventral. Ventral. Because we tend to get confused which is dorsal, which is ventral. Dorsal meaning is the back side. Okay. Imagine this is the lungs. This part, this part is dorsal towards the back side. Okay. Towards the back side, this is dorsal. So back side is dorsal and front side is ventral. Okay. So, if you're talking about dorsal and ventral positions, we can say that dorsally, what is behind the lungs? What is behind the lungs? Obviously, it is vertebral column. So, obviously, dorsal vertebral column meaning we can check the options. It is option B. And sternum. Sternum comes in the front. So, we can't choose option A because dorsally it is covered by Dorsally, it is by vertebral column, and ventrally, it is by sternum. Okay. Now, what is laterally? Laterally, we have ribs. Okay. Laterally, we have ribs. This is a line in NCRT. Laterally, we have ribs, and inferiorly, we have diaphragm. Okay. Laterally meaning towards the sides, and inferiorly means towards the lower side. Okay. Now, next question. What forms the lower boundary of the thoracic cavity in humans? This is very easy. I just told you now. Laterally, it's the ribs. So, lowerly, it's the dome-shaped diaphragm. Okay. Now, the next question. Double-layered membrane that reduces the friction on the lung surface is known as option A, pericardium, option B, peritoneum, option C, pleura, option D, meninges. So, lung is covered by this double-layered membrane called pleura. Okay, pleura basically what happens is there's an outer parietal pleural line on the thoracic wall. Okay, and the inner visceral pleura covers the lung surface. Okay, so there's this outer pleural wall, there's this inner pleural wall, and between there's a pleural fluid. Okay, this pleural fluid. Okay, this decreases friction during expansion and contraction. Okay, now what is the main function of the conducting part of the human respiratory system? What is the main function of the conducting part of the human respiratory system? What is the main function of the conducting part? Diffusion of gases into the capillary blood capillary beds. First thing you need to know is the conducting zone does not take part in gas exchange. Okay, so wherever it's written gas exchange, diffusion, directly cancel it out. Direct active transport of bicarbonate ions. Do you think that is possible? No. Generation of metabolic heat during expiration? No. Basically, what happens is the conducting part, okay, its cells basically secrete mucus. Cilia is used to trap dust and foreign microbes. Okay, it adds moisture to avoid humidification. Basically, the lung desiccation. Okay, it thermoregulates the air to the normal room temperature or the human temperature. Okay, 37 degrees Celsius before it reaches the alveoli. Alveoli is fragile. Okay, so before that, it should be brought to the body temperature. Okay, with this we finish section 1. Okay, next section is pulmonary volumes, capacities, and spirometry. This is very important. This is the most fun where the questions keep repeating. So we'll be continuing with this. Tidal volume is the volume of air inspired or expired during normal respiration. In a healthy human male, this value is approximately. Basically, what is tidal volume? Tidal volume is basically the volume of air moved during quiet resting ventilation. We should always know in adults there's a standardized value of approximately roughly 500 ml. Okay. Now, a healthy human can inspire or expire approximately how much air per minute during normal breathing. First, we need to check. Minute ventilation is calculated by multiplying tidal volume with healthy respiratory rate. So let's take a healthy respiratory rate. What is the limit? What is the healthy respiratory rate? 12 to 16 breaths per minute. Okay, this is the lower limit. This is the upper limit. So the lower limit we do 12 into 500. 500 is the tidal volume. Okay, okay, 600, 6000 ml per minute. Upper limit, how much? 500, 16 into 500, 8000 ml per minute. Okay, so this is the answer. The additional volume of air that a person can inspire by forceful inspiration is called what? See, there's this 2000 to 5000, 2500 to 3000 ml, okay, that of air that remains, okay, the additional volume that a person can inspire even after taking the normal inspiration. This extra 2500 to 3000 ml is called as inspiratory reserve volume. Okay. Sorry. What is the typical clinical range for expiratory reserve volume in a healthy human adult? Okay. See, ERV is the maximum additional volume. Okay. That can be forcefully expired after a normal tidal expiration. Okay. The normal value basically ranges from 1000 to 1100 ml. Okay. So till now, what did we learn? The values tidal volume we heard 500 ml. IRV we learned as 2500 to 3000 ml, and ERV we learned it as 1000 to 1100 ml. Okay. So we learned the three basic ones. The volume of air remaining in the lungs even after a forced expiration is designated as. Okay. So after there's a remaining air left behind after forceful expiration, that is known as residual volume. Basically, what happens is even after this, lungs cannot never be completely deflated. Okay, and this residual volume is usually almost about 1100 ml to 1200 ml. Why cannot residual volume measured by standard spirometer? See, first thing we need to know is spirometer works by measuring the volume of moving air entering or exiting an open mouthpiece because residual volume stays trapped inside the lungs to prevent alveolar collapse. Okay, it exits and into the spirometer. Okay, what happens is the capacities that include RV. What are the capacities that include RV? We have FRC and we have TLC, which I'll be explaining to you. Okay, they cannot be directly measured by spirometer. Why? Because how does spirometer works? It measures the volume of moving air entering or exiting through an open mouthpiece, correct? So if RV stays trapped inside the lungs, how can it never exits into the spirometer tube, correct? Then how can we measure it? Correct? What is FRC? FRC is basically functional residual capacity, correct? What is TLC? TLC is the total lung capacity. It is everything together, added everything together. Okay. Now we'll be learning. So, what is the answer for this question? Why cannot it be measured? Because it is never forcefully expelled from the respiratory. Now, what is inspiratory capacity? It's the maximum total volume of air in an individual that can inhale following a normal unforced expiration. So, how can we answer this? It is basically TV plus IRV. Okay, so that is the answer. TV plus ERV? No, TV plus IRV. How can it be IRV plus ERV? No, wrong. TV plus IRV plus ERV. No, ERV should not be included. Okay, expiratory capacity. Expiratory capacity. First thing you know, expiratory capacity, ERV will be there. Okay, so IRV option cancelled, TV plus RV cancelled. So it's either residual volume plus or tidal volume. Obviously, it's tidal volume, correct? Basically, it is the maximum volume a person can exhale after a normal tidal inspiration. Okay. Next question. Functional residual capacity is the volume of air that remains in lungs after a normal expiration. It equals TRV plus ERV. Sorry for that. TV plus TV plus ERV, ERV plus RV, IRV plus RV, TV plus IRV plus ERV. So, what is functional residual capacity? I just told it to you now, correct? Basically, after a quiet exhalation, after a passive exhalation, the lungs still contain both the air you could blow out forcefully and the air that cannot be blown out. The air that can be blown out forcefully is ERV, correct? And the air that cannot be blown out is RV. So, what is it? So, it is ERV plus RV. That is FRC. That is the answer. The maximum volume of air a person can breathe in a forced expiration is known as. This is a direct answer. It basically means vital capacity. So, what is vital capacity? Basically, the maximum air movement possible in lungs. Okay, it presents the volume of air you can inhale after a maximal forced exhalation or exhale after a maximal forced inhalation. Okay, so what is the answer? It is vital capacity. Express vital capacity using its correct pulmonary components is basically this is direct question ERV plus TV plus IRV. Basically, what it brings together both ERV and IRV. Okay, and it gets tidal volume together. Okay, this is a very direct question. Total lung capacity is the total volume of air that can be accommodated at the lungs at the end of a forced inspiration. It can be written as first we have to write TLC. TLC sums to the total air that the lung can hold, correct? So first we need to know total lung capacity is ERV plus TV plus IRV plus RV. What did we learn this as? This is basically VC plus RV. So we can write this as VC plus RV. Okay. Next, we go on to section 3. pulmonary mechanics, and intrathoracic gradients. Okay. First question. Inspiration occurs when the intrapulmonary pressure is higher than the atmospheric pressure, lower than the atmospheric pressure, perfectly equal to the atmospheric pressure, twice the atmospheric value. First, you should know that twice the atmospheric value is a option that is not possible. It doesn't make sense. Okay. Basically, when fluids move down the pressure gradient, that is when the air enters from for air to enter the lungs from the atmosphere, the pressure in the lungs should be dropped below atmospheric pressure. Okay. What does this cause? This causes a transient vacuum. Okay. This causes an intrapulmonary pressure. Okay. This pressure inside the lungs, this intrapulmonary pressure should be dropped. So if it should be dropped, what should it be? It should have lower than the atmospheric pressure. Correct? Next question. Contraction of the diaphragm increases the volume of the thoracic chamber in which specific anatomical axis? Dorsoventral axis, anteroposterior axis, lateral axis, oblique plane. See, when the dome-shaped diaphragm contracts, no, it flattens towards the abdomen. So when it contracts, it flattens towards the abdomen. So this is the abdomen. It flattens like this. Okay. So this structural movement basically expands the vertical dimension of the thoracic cavity. So which axis is it? Can it be lateral axis? No, it can't be oblique. Either it should be dorsoventral or anterior posterior. Can the lungs go dorsoventrally? Does that make sense? No. It will be anterior posterior, correct? Anterior to posterior axis, correct? Now, the contraction of the external intercostal muscle lifts up the ribs and the sternum, causing an increase in the volume of thoracic chamber in the anterior posterior axis, dorsoventral axis, infralateral plane, longitudinal axis. This is something now if we know about anterior posterior axis. So obviously it is going to be dorsoventral axis. We are talking about sternum here. Okay, causing an increase in the volume of the thoracic chamber. Okay, obviously it's going to be dorsoventral. So basically, when external intercostal muscle pulls the ribs up, okay, and outward like a bucket handle, it pulls like a bucket handle. Now it looks like this. Okay, so expanding, it does an expanding to the front to back clearance of the chest cavity. Okay, so basically it becomes dorsoventral. Okay. Expiration is initiated when relaxation of diaphragm and external intercostal muscle causes an increase in the thoracic volume, a decrease in the thoracic volume, increasing interpulmonary pressure above atmospheric levels, immediate closure of glottis, active transport of nitrogen into alveoli. See, we need to know that the normal quiet expiration is a passive process. So when the diaphragm and the external intercostal relax, no, the elastic tissue of the muscles recoil. Okay. The thoracic volume decreases. Okay. And this compresses the gas inside, raising the pulmonary intrapulmonary pressure. It can above the atmospheric level. So it can be plus 1 to 2 mm Hg, driving the air out. Okay. So there should be a decrease, right, in the thoracic volume. This compresses the gas inside, correct? So there should be a decrease in thoracic volume, increasing the intrapulmonary pressure above the atmospheric levels. Okay. Humans have structural capability to increase the strength of respiration using muscles located in A. Neck, B. Abdomen, C. Pelvis, D. Thighs. Thighs, pelvis directly cancel. You think the strength of respiration muscles are there in neck? No, it's in the abdomen. Okay. So Breathing relies on diaphragm and external intercostal muscles. Okay, they can increase the depth. They can increase the depth and force of ventilation by recruiting accessory respiratory muscles, and they are located in neck. But they are located in neck. But the main muscles are in abdomen. Okay, during deep forced expiration, which muscle contracts actively? Now, forced expiration is an active process. Okay, so which muscle do we need? Internal intercostal muscles, correct? And abdominal muscles. Abdominal muscles contract the drive of the visceral organs against the diaphragm. Okay, and this causes the shrinking of the thoracic cavity. What does internal intercostal muscle do? They depress the ribs down, correct? So what is the answer? Internal intercostal muscle and abdominal muscles. Now the next section we are going into is gas exchange and solubility loss and partial pressure. Pressure contributed by an individual gas in a mixture of gases is known as. First, we know that in Dalton's law, okay, there's something known as individual pressure contribution of a specific gas. This is partial pressure, okay, PO2 or PCO2. How can it be total pressure? No, individual gas. We're talking about individual gas. It can be partial pressure, PO2 or PCO2, where P represents the partial pressure. Atmospheric index, no. Osmotic tension, no. Okay. Now, what is the value of partial pressure of oxygen in atmospheric air and in alveolar, respectively? That's a good diagram in NCRT. That's a very good diagram in NCRT that we need to refer. If we know this diagram properly, we can know this question. Okay. So, what is the value of partial pressure of oxygen in atmospheric air and alveolar, respectively? In alveolar, it's 104. So if you know that answer, you can answer, easily mark this answer. Okay, 104, option A. Okay, so basically, in dry atmospheric air, okay, it is 159 mmHg. Okay, and in alveolar, it is 104 mmHg. Okay, the partial pressure of carbon dioxide in deoxygenated blood. This is also a direct question in deoxygenated blood. This is also based on the diagram. It is 45 mmHg. So please study that diagram. Okay. Please study the diagram. It is very important. Okay. Once I'll be doing a revision class. Okay. Just the diagrams of NCRT. So please subscribe. Okay. I'll be teaching all the diagrams, all the important diagrams I'll be going through. So I'll go through once the revision continues. As the revision progresses and finish at least one round of revision for this, we'll go through the diagram revision also. Okay, so that time I will revise this. So again, please go through these diagrams daily. They give you a idea and don't let you forget. Okay, so the next question. It's the same question based on the diagram. What is the correct value for partial pressure of oxygen within the systemic oxygenated levels? It is 95 mmHg. Okay. PCO2 in alveoli and inside the working tissues respectively. What is it in alveoli? Okay. In alveoli, PCO2 is 40 mmHg. Okay. And inside working tissue, we just learned now, it is 45 mmHg. 104? No. 40 mm? No. 40 to 45? Yes. So option A. So basically, alveolar air has 40 mmHg. Okay. Metabolic active tissue cells, they constantly synthesize CO2. So they have to maintain an intercellular PCO2 of 45 mmHg. So the gradient, there's a gradient. So basically, the gradient ensures that the CO2 diffuses out of the tissue into the blood and from the blood into the alveoli. Okay. Next question. Why does carbon dioxide diffuse across the respiratory membrane much faster than oxygen despite smaller partial pressure gradient? Because its molecular weight is lower, because its solubility of CO2 is 20 to 25 times much higher than that of oxygen, because it utilizes primary ATP pumps, because it travels through open lymphatic channels. Why is it? Because the solubility of oxygen compared to carbon dioxide, meaning the solubility of carbon dioxide compared to oxygen is 20 to 25 times higher. Okay. The gradient of oxygen is large. What is it? 104 minus 40 mmHg, which is 64 mmHg. Okay. While the gradient of for carbon dioxide is small. Okay. Carbon dioxide is 45 to 40. So 5 mmHg. So CO2 is what? 20 to 25 times more soluble in water and plasma than oxygen. So it diffuses more rapidly across the respiratory membrane. So what is the answer? Because its solubility is 20 to 25 times higher than oxygen. Now, next question. The diffusion membrane consists of three major layers. Which of the following is not done? Thin squamous epithelium of alveoli, endothelium of alveolar capillaries, substance composed of thin basement membrane support, ciliated columnar cells of the primary bronchi. See, the diffusion membrane should be thin. Okay, it should be lesser or equal to 0.5 mm. Okay, why is this? This is to facilitate the gas exchange. Now, this is composed of three layers. What are the three layers? We have the thin squamous epithelium of the alveoli. Okay, we have the endothelium of the alveolar capillaries, and the thin basement membrane. Okay, where the ciliary columnar cells, where is it? It's in the upper conducting tract, right? So that's the answer. These three can be cancelled. Okay. Now, with this, we go to section 5, hemoglobin kinetics and gas transport mechanics. So, under normal conditions, what percentage of oxygen is transported via the chemical form of oxyhemoglobin within RBCs? So oxygen transport occurs within two routes. 97%, what happens is it chemically is bound to iron containing pigment, okay, which is known as hemoglobin, and it is within the RBC. While there is the remaining 3% that is dissolved in liquid plasma. Okay, so which is the option? Option number D. So now I told the remaining 3% of oxygen does not bind to hemoglobin, is transported through what? Dissolved in blood plasma fluid. Each molecule of hemoglobin carry a maximum of how many molecules of oxygen? 2, 4, 6, 8. This is directly we know. One hemoglobin, 4 molecules of oxygen. Okay, so 4. Binding of oxygen with hemoglobin is primarily related to, first thing we need to know, it's not related to total plasma index, not related to the absolute blood volume. How can it be related to the partial pressure of nitrogen? No, directly it is related to the partial pressure of oxygen. Okay, the mathematical graphical representation obtained when the percentage saturation of hemoglobin with oxygen is plotted against PO2 is called as what? Is it called J-shaped? No, it's called sigmoid, correct? Sigmoid oxygen dissociation curve. It's not hyperbolic. It is not a linear clearance plot. Now, which of the following physiological steps occurs in the alveoli to promote the synthesis of oxyhemoglobin? In alveolar, environmental conditions favor loading oxygen onto hemoglobin. So these conditions include high PO2, correct? High PO2. So low PCO2, low H concentration meaning more the pH and low temperature. Okay. Together they cause what kind of curve? They cause a left shift. Okay, in the curve. So there's an increased oxygen affinity. Okay. At tissue level, dissociation of oxygen from oxyhemoglobin is triggered by which environmental mix? So if there is a dissociation from oxygen, we need to look at low PO2, correct? Low, so high cancel, so low PO2. We can take low temperature low alkaline. Yes. Why? No. It is for synthesis, correct? So low PO2, high PCO2, high concentration, which is acidic, okay, and higher temperature. Okay. Now, every 100 ml of oxygenated blood, systemic blood can deliver around how much oxygen to human tissues under normal resting physiological conditions. So, what is the answer? See, under resting physiological parameters, 100 ml carries about 20 ml of oxygen. So, when it passes through resting tissues, it returns to the heart carrying approximately 15 ml of oxygen. That means how much oxygen is delivered to the blood flow? 5, correct? This 15 ml is brought back. 5 ml is gone. So obviously 5 ml is utilized. So the answer is 5 ml. Carbon dioxide binds directly with the amine groups of hemoglobin to form the complex known as carbo carbaminohemoglobin. Correct? So carbon dioxide interacts with the protein globin portion of the hemoglobin. Correct? And it rather than the ion binding heme site. So it chemically bonds to the chemical state termed as carbaminohemoglobin. Okay, and occurs when carbon monoxide binds with hemoglobin. So what's the answer? Carbaminohemoglobin. What percentage of carbon dioxide is transported through blood system bound as carbaminoglobin? Hemoglobin, sorry. So CO2 relies on three pathways. 20 to 25% it moves as carbaminohemoglobin in RBC. Okay, carbaminohemoglobin. Okay, 7% directly as plasma, dissolved in plasma, and 70% as bicarbonate ions. So obviously they are asking here is carbaminohemoglobin, so 20 to 25%. The vast majority of carbon dioxide produced in tissue metabolism is carried in the body as bicarbonate ions. Name the zinc containing enzyme formed in high concentration within the RBC that catalyzes the interconversion of carbon dioxide and water into bicarbonate. What is it? Carbonic anhydrase. It is a metal enzyme, okay, which uses zinc as the cofactor to accelerate the conversion of dissolved CO2 and water into carbonic acid by several orders of magnitude. So what is it? Carbonic anhydrase. At tissue site where PCO2 is high, carbon dioxide diffuses into the RBCs and undergoes which reaction sequence? It remains trapped as gas blocks. It forms H2CO3 which dissociates into HCO3 and H+. It condenses into solid urea components. It binds immediately to blood platelets. Directly we know it does not bind and condense into solid urea components, and it does not bind to blood platelets. Does it remain trapped as gas blocks? No. Basically, it forms H2CO3. And this H2CO3 basically dissociates into HCO3 minus and H plus. Okay. So in carbonic anhydrous, if this is carbonic anhydrous, H2CO3, okay, and then it dissociates again here as HCO3 minus plus H plus. Okay. So at high PCO2, CO2 drives into the blood, drives CO2 into the RBC. Carbonic anhydrous forms the formation of, catalyzes the formation of carbonic acid, which dissociates into bicarbonate ions and hydrogen ions. Now, every 100 ml of deoxygenated blood reaching the alveolar capillaries unload how much carbon dioxide into alveoli? This is direct question. So every 100 ml of returning deoxygenated blood delivers 4 ml of CO2 and 5 ml of O2. So they are asking here CO2, deoxygenated blood, carbon dioxide. So 5 ml of oxygen and they're asking carbon dioxide. So 4 ml of carbon dioxide. Okay, so now we're going into the last part of the topic, neurological control and chemical feedback loops. The primary respiratory rhythm center responsible for regulating human ventilation is located in. So automatic breathing is driven by respiratory rhythm center. Okay, by a cluster of neurons located in medulla oblongata in the brainstem. It establishes the baseline pace of inhalation and exhalation. So what is it? Medulla oblongata. The pneumotaxic center can moderate the functions of respiratory rhythm by shortening the duration of inspiration. It is located by C. The answer is Pons region of brain. Correct? It can't be. It's not medulla. Okay, it's not thalamus. It's not midbrain. It is pons. How it is pons? This pneumotaxic center. Correct? Okay. It is located in dorsal pons varoli. Okay, this regulates the medullary rhythm center. So it acts like an off switch for inspiration. So by signaling an early termination for inspiration, it shortens the breath duration. Okay, which increases overall respiration rate. Now, the chemosensitive area adjacent to the medullary rhythm center is highly sensitive to accumulation of, obviously it's carbon dioxide and hydrogen, correct? Oxygen and nitrogen, no. Carbon monoxide and no. Glucose and insulin, not at all. Correct. What happens when the central chemoreceptors, okay, they do not monitor oxygen. They monitor concentration of carbon dioxide and hydrogen, okay, in the CSF. So when they accumulate, there's an increase in ventilation and they try to eliminate the excess gas. Peripheral chemoreceptors capable of changes in arterial blood, PCO2 and H plus are located in. See, they're not located in jugular veins, not in hepatic portal veins, pulmonary veins, no. It is located in the aortic arc and the carotid artery. So the peripheral chemical monitoring, they rely on the specialized sensory bodies, aortic bodies. They are located in the aortic arc wall and carotid bodies, which are the bifurcation of the common carotid arteries. Okay, they send afferent signals to the medulla with the arterial PCO2 rises. Okay. What role does the concentration of oxygen play in the feedback modification of human respiratory rhythm center? Highly significant and primary, quite insignificant under normal parameters, directly proportional to tidal volume changes, controls pneumotaxic speed completely. Basically, concentration is very insignificant during normal parameters. We usually look at carbon dioxide and hydrogen, correct? So, regulatory role of oxygen is insignificant. Respiratory range changes long before arterial oxygen drops to critical level. Okay, so it is quite insignificant. Asthma is a clinical condition. So now we are going to clinical disorders and occupational lung diseases. Asthma is a clinical condition characteristic by difficulty in breathing and wheezing, caused by bacterial infection. No. Inflammation of the bronchioles and the bronchi. Correct. Progressive reduction of the number of alveoli. No. Deposition of calcium inside the diaphragm. No. Okay. So basically, it's an inflammatory disease of the conducting areas. Okay. It's a hypersensitivity reaction. You'll be learning about hypersensitivity reaction when you get into medicine. You'll be learning about the types of hypersensitivity reaction. Asthma comes under type 1 hypersensitivity reaction. Okay. So localized inflammation, spasms of the smooth vessels, okay, narrowing of the lumen, producing characteristic high-pitched wheezing sound during exhalation. Okay. Emphysema is a chronic disorder in which respiratory exchange surfaces is severely reduced to damage due to alveolar walls, epiglottis muscular walls, bronchial cartilage, trachea. No, it is not trachea. It can't be bronchial cartilage also. Alveolar walls, correct? It's a damage of alveolar walls. Basically, what happens is it's a progressive obstructive lung disease. Okay, where chronic irritation leads to the permanent breakage of the alveolar wall. It breaks down. So what happens is this individual alveoli fuses into this less elastic air pockets. Okay, reducing the total surface area available for gas diffusion. Now, what is the major environmental or lifestyle cause of clinical emphysema? Air pollution from stone crushers, cigarette smoking, consumption of contaminated water, genetic deficiency of insulin receptors. See, prolonged cigarette smoking is the leading cause of emphysema. Basically, what happens is this inhaled toxins trigger the release of protease release like elastase. What happens is when elastase is released, it causes the degradation of the structural proteins of the alveolar walls and it destroys the lung elasticity. So it is cigarette smoking. Occupational disorders like silicosis and ab- abscessosis are common among individuals working in which industry? Dairy and poultry? No. Stone breaking, grinding, and mining. Correct? Textile spinning? No. Chemical? No. Long exposure into dust and grinding causes inflammation of lung tissues, leading to proliferation of fibrous tissue. This is known as fibrosis. Okay. What clinical step is recommended for workers prone to occupational diseases to prevent long-term lung damage? Daily consumption of calcium supplements? No. Wearing protective mask on site? Yes. Wearing protective mask and protecting yourself from these dust is the best way you can do to protect your lungs from long-term damage. Reducing their daily tidal volume. What is this? No. Taking periodic antibiotics because it's not a bacterial disease for us to consume antibiotics. Correct. So with this we finish respiration. I'll be doing a part two very soon by if possible today only, and I'll be uploading it. Thank you so much. I hope this was very helpful for you and you covered the entire syllabus for respiration and you revised thoroughly. If you have any doubt, put it in the comment box. I'll answer as soon as possible. And I'm just developing this channel right now, so I'll be soon creating a Telegram channel where you guys can come and you can ask me doubts, and I'll be sharing the materials so you guys can go through and everything. Thank you for staying with me. I hope you enjoyed the session and you learned a lot. Thank you so much.